Really grateful to be here, and the goal of today is to tell you a story about these worm blobs. The lab, as uh, was described, essentially the way we think about this is doing curiosity-driven science. And we're a bunch of physicists, engineers, mathematicians, and we look at organisms as living, as nature's engineers and nature's physicists. And we recognize that they solve some interesting problems. And we watch them and learn from them and try to understand how they solve some interesting problems. And our lab kind of adopts the Krog principle in spirit, which basically says that for every question there is a system in which it can be most easily studied, just like we wouldn't study flight in C. elegans or mice. Effectively, for each question, there is a proper organism to study it in. So we're very promiscuous kind of in our organisms of choice and study everything from the single cell ciliates that can generate motility using non-canonical fuel, if you will. Instead of using ATP, they use calcium. Um, from these to developing protein networks and single cells to organismal level questions from how we blink or how flamingos are able to filter feed in complex hypersaline, muddy environments, all the way to collectives of cells, how they may talk to each other across long distances just using hydrodynamics, or a team of dogs, how they achieve some task in harsh environments such as Antarctica. And today's story is, of course, on these fascinating living systems, these worms that tangle up together. So this is one part of the lab, and the other part of the lab is where I redeem myself as an engineer and actually do stuff that is useful to society. We think of it as frugal science and developing hardware to democratize these tools so that we could do diagnostics under a tree, to science deep in the Amazon rainforest, and even enable high school students in my own backyard in Georgia and Alabama to be able to do hands-on science, because hardware is both an enabler and an impediment, and oftentimes the cost of hardware is what distinguishes between who gets the opportunity to do science and who gets left out. We're all very lucky over here, and all of you probably scientists, because you have excellent instruments to do with, and our goal is to ensure that the billions of people we share this planet of ours with get the same access to hardware. For the graduate students, postdocs, I also run this jungle biomechanics lab in the Amazon rainforest for the last few years. Essentially, we take students, we have an application, I think it'll open up in a few months if you go to our website, and we take students to the Amazon for a few weeks to nurture and ignite their curiosity, to take a walk in the woods and essentially practice this following Eisner, who said that the urge to explore can be discovered, like the ability to discover can be developed, and you kind of give people a chance to work on non-model systems. And here's an example. This is a theoretical physicist, Avanish, from Princeton and now at Stanford. He, when he took a walk in the woods, saw these beautiful caterpillars on a tree exhibiting emergent motion and active mimetics. And we have an abstract submitted on this for the Global Mars Summit, in case you're interested. But essentially, that's how it goes. We walk, make discoveries, and start to think about systems. So that's a good segue. By the way, I'm very open to being interrupted. I think 2 weeks ago, I was at a physics talk, and they didn't let me go past the second slide. So I feel like I've already given you a lot more. So feel free to interrupt me, and I'm happy to answer any questions. Essentially, when people think about collective active matter or emergent properties, physicists typically start thinking about colloidal-like or spherical-like systems. You may think of them as bees or penguins huddling, or colleagues who study fire ant troughs. And a perspective here is, if you stretch the aspect ratio of the individual agent, you start to see interesting geometrical behavior. So you might see the caterpillars I just showed you, all the way to, if you really, really stretch the aspect ratio of these, you get these polymer-like filaments, and these are the worm blobs. And so the question is, what happens as you stretch the individual's aspect ratio? And so one way we've tried to rationalize this is to think about a linking number. And I'm going to show you in this plot, essentially think of this as the aspect ratio, so geometrical complexity, and essentially how they link, so topological complexity. And examples help. So for example, you may have rigid filaments that may have a lower linking number. And this may include examples like thinking about the emergent structural stiffness, rigidity that may arise in bird's nest. And if you increase the linking number a little bit by creating a little bit more entanglement, you may make a structure out of staples, or think about it as Dan did with a bunch of these staples. You get these emergent materials, and you can further increase the linking number by increasing the number of touch points to create fire ant troughs or even robots made out of robots. And you start to see that there is the ability to harness this topological complexity. This is where you reach the final quadrant, where you increase both the geometric complexity and the topological complexity, where the worm blobs lie. And recently, there has been some interesting work as we want to think about these stochastically tangling robotics. So here's an example where these robots are not programmed with anything, but they emergently adapt to the object through their stochastic tangles. And so this is just early work, and kind of the question is, nature gives us some examples on how to harness this topological geometric complexity to do useful things, and what can we learn from them. The reason, at least I care, is it would be interesting to make kind of these geometrically and topologically complex structures. Let's refer to them as blobs. And typically in science fiction, they're oftentimes scary things. So this is an image from Stranger Things, where you can imagine these structures that are tangling and active. They may slide around, fall off a table, slide under a door, maybe even fuse together. And although this is often the stuff of nightmares, I think this could be an interesting technology, and there might be some positive things. But my thesis today is that the reason we can't build these things is not because we lack the ability to design materials. In fact, over the last 60 years or so, we made a lot of advances in thinking about this field of soft matter and be able to design polymers and hydrogels, such that as we evolve as societies, our understanding of the materials, whether it's in the Stone Age or the Iron Age, informs the way we think about the robots and the structure we make, such that today we dream about these robots and structures not made out of rigid things, but out of flexible systems. But we still lack basically a knowledge in how to put these things together, which is very aptly captured by this P.W. Anderson's kind of thesis that more is different. And so hopefully over the next 30 or 40 minutes today, I'll kind of offer you that the reason we may not be able to do this is because emergent dynamics in these systems is tricky, and we might be able to use living systems as a way to understand kind of the physics. And so that's the thesis for the next minutes, is that hopefully I'm out to convince you that worm blobs are a tractable model system to study tangle active matter, and then perhaps they may inspire kind of flexible soft robots. So that's kind of where I am in, and I'm going to tell you kind of the genesis of the story and how I stumbled upon these worms. So I did my PhD at Stanford, and for those of you who've been there, you can see this is the Lake Lagunita, and there's the Hoover Tower in the back. And California at that time was going through a drought. So this lake I'd never seen during that time filled with water. But I did continue to stay on to do my postdoc, and one day it rained. You have two perspectives on this. If those of you from California, you know that when it rains, everybody grumbles because we expect 365 days of sunshine. But all the undergrads, grad students, we were also excited, and everybody started to bring out kayaks and wanted to enjoy this lake. And I stole a pipette and a water bottle and went to take a walk around this lake, asking a simple question: what life emerges in this ecological niche which has been dry and barren for half a decade? And I didn't expect to find much, but I saw this, and this blew my mind away. And I did what any of us would do. I took a stick and I poked at it. And to both my delight and my then girlfriend's horror, it was alive and it moved. And this is essentially a worm blob where the water has receded. And these are some of the worms. In some of the areas, the water hasn't receded, and they're quite happy and living in detritus. You can see I'm going to bring a pipette, and I'm trying to suck some of them away, but they're Houdinis, and they go back into the wet granular matter very, very quickly. But this is how they kind of live. These are aquatic oligochaetes. And so I knew I had found my first project when I set up my lab at Georgia Tech. So this is Yasmin, who was then a postdoc with me, and Dan Goldman, and now a faculty at Notre Dame. This is the protagonist of today's story. This is California blackworms, or Lumbricus variegatus. They're really, really highly anisotropic, flexible active filaments, and they're 0.5 millimeter in diameter and 3 centimeters in length, so aspect ratios of greater than 100. And they're oligochaetes. Oligo means few, and chaete means hair. They have a few hairs along their body that help them in locomotion. This is Yasmin during the pandemic when everybody was baking bread. She's playing with this living dough. This is 50,000 worms. It's just worms and a little bit of water coated around them. And you can start to see that it flows between her fingers. And it's like living silly putty. And if you were to drop it, it would still maintain its shape. It wouldn't bounce, but it's a cohesive, tangled material. It's you can make a small blob, you can make a big blob out of these things, and you can write the name of the university or the name of the journal you want to submit this paper to. And I can tell you from experience, it does not change the editor's mind if you write their journal name. So it does not work. Let me pause here and see if there are any clarification questions so far. Is everybody with me? Anything I can answer right away? Yeah. Excellent question and great segue. In one slide, I'll answer it. So, those, Yasmin, as you correctly observed, these are aquatic worms, and this is how we culture millions of them in our lab. They live quite happily in this granular matter with their heads down and their tails up, but they will blob both in water as well as outside, which leads to the question of why do they blob? It turns out nobody knew, and this was kind of the first question we asked, and I think is all on your minds: why do you blob? And so we had a simple hypothesis that you're taking this fish out of water, if you will. Perhaps it's a strategy to reduce evaporation. And here I'm showing you a side view and a top view of a worm blob placed on plastic. So now it's in air. And you can start to see that the worm blob will, spontaneously kind of break symmetry, shoot out an arm to stretch. And if it doesn't find any moisture or hydration, it will retract back and form this blob. But what we were able to appreciate is that it forms this hemispherical structure you can start to see from the side view. And that kind of led us to this hypothesis of evaporation reduction because it's forming this beautiful geometrical structure. And Yasmin did beautiful experiments with setting up this camera and doing repeats. And essentially what we're doing over here is plotting the 2D projected area as a function of time. And here's the kind of intuitive takeaway. If I'm a single worm, I die in a few minutes. But if I'm that same worm in a blob of 1,000 worms, I'm still alive for hours. Effectively, it reduces my evaporation loss of being in a blob because it protects me from evaporation. And you can start to think about each of these worms as essentially a cylinder. And it's a packing problem of how many cylinders can you fit to create this surface to volume area reduction kind of structure. And that's kind of what the simple theory and experiment is. So this is kind of like a vanilla hypothesis that indeed they're forming the structure to reduce hypothesis. Let me pause here. Everybody okay with this? Is this clear so far? Okay, so no surprises here. That's a good question. We think it's just desiccation. They're evaporating. So
could you say that again?
I updated my watch, so now this has Apple Intelligence. It's way too smart. Because they're aquatic, they are always coated with a little thin film of water, similar to how you get out of a shower, you're hydrated. As they dry up, they'll shrivel up and die. So we think some kind of hydration response. Is that what you were asking? Going back to your first encounter with the dog. Did you found them in a small puddle that did not dry up, or both? Had they resuscitated after a spell of dry? So because it's an ephemeral kind of dry area and water is evaporating by the time I'm going there, it's at the edge of where the water has already gone in the ground or evaporated, but you can also find them in the shallow areas. And I'll show you some more examples after floods. So they're available in both areas. Of course, they're much more balled up where all the water has gone away, but they're more individual or in groups. What other? Uh-huh. Exactly. Good question. And yeah, I'll show you some videos that we are starting to tackle in foraging collectively. Yes. Beautiful observation. It's basically a topologically restructuring system. It's not static, and essentially they're recircling because the ones on outside presumably get desiccated, and how they do this through reptation is unclear, but very good observation. And so what we realize is this strategy of blobbing is actually quite common across different worms. So here's some colloquial observations after Texas floods where the water has receded. These are sewer worms called tubifex, and they also blob. And then finally, one of my most favorite videos, we've done some field work down in this cave. I'll let Caver Dave explain to you.
Sulfur Cave is full of toxic gases. If you're going into Sulfur Cave, can't go in there without self-contained breathing apparatus. Just like a stinky, muddy, goopy hole in the ground and deadly to humans and most other creatures. That's full of life. My initial reaction when I first saw the worms was, wow, there's really something living in this cave. And it immediately made me think they could be a new species that probably live nowhere else on the planet. To keep these worms alive between the cave and the museum, my homemade method of oxygenating these worms is just to aerate them through this little straw.
Okay, so this is not how we culture our worms. We have a proper setup, and this is not how my graduate students keep worms alive. But nonetheless, it offers that this might be a survival strategy against harsh environments, whether in sewers or sulfur caves. So this is where Yasmin showed me an experiment that really surprised me and challenged my intuition. Here's essentially, she's brought a baking dish from Walmart. You can see the US flag right there. So we're supporting made in USA, or I don't know, maybe it's not. And then essentially this side is cold and this side is hot. As you can see, there are nucleation bubbles. This is a little bit hot. And it's under bright light. And if she places the worm blob in the center, the question is, well, what will happen? And she discovered this was remarkable, is that this amorphous tangled structure exhibits this kind of negative thermotaxis moving towards the cold side. And I didn't expect this because how does the worm know? How does the worm blob know? Here's another experiment to show you that there's a very dynamic rearrangement and somehow they are able to collectively move. And there is obviously, as you can see, survival in being in a collective, because if you're individual and you stochastically move to the wrong side, you will die. Yeah. Controlling for the humidity, because based on the condensed water, the cold side is not. Controlling for all of this is in water. It's in a thin film of water. Remember, those are bubbles. So it's all happening in a shallow dish of water. So there's a thermal gradient in the water. Yeah. And so they blob in water and they can locomote in water as well as in air, as I've shown you. Very multifunctional and cross domain. What's the temperature difference? Good question. It is about 9 to 11 degrees. So the left-hand side is cold over here is about 18 to 16 degrees. And then 27 to 28 degrees on, or Celsius. So they will die after the temperature reaches. So some of these spots are actually quite hot. So maybe 10 to 11 degrees delta T is probably less. There might be maybe twice that or something. And so to understand how these actually move, it was very hard to see this in this large blob from the bird's eye view. So Yasmin and I came up with a simple experiment where we created almost like a Hilly-Shaw cell, and we're putting small worms. And what we start to see is that in these about 12 or 15 worms, is that we started to observe that the worms started to stretch towards the cold side, which we call now puller worms. And the worms in the back started to coil up and started to lift the center of mass as if to reduce friction. So we call them wiggler. Essentially, we developed a hypothesis that the worm is able to move this because of binary differentiation of these tasks of pulling and kind of wiggling to reduce friction. Now, to test this, we turned to simple robotic structures in collaboration with Dan Goldman. And here, essentially, instead of temperature, they're responding to light. And these are robots that will kind of flap their arms when we call this as a crawl gait when it's towards light. And when it's towards lower light, you can start to see that they will do a wiggling motion. This is kind of the binary differentiation we're programming in a simple model. And we can cover these up with plastic, kind of these mesh-like structures, so we can make kind of a smarticle blob or a slob. And here's how we're testing this hypothesis. So Yasmin's put light over here. And essentially in this video, if only the bottom worms wiggle to reduce friction, you see that the entire structure doesn't move. And if only the front robot starts to pull, it cannot generate enough force to pull the entire blob towards the light side. But if both of these things start to happen, which is exerting a pulling force and reducing the friction, you can start to see we get net locomotion here. And so this is a way we rationalize kind of this thesis. And oftentimes I get criticisms that these don't look like worms. And essentially the response here is this is a model and all models are wrong, but this is useful and helps us test kind of thesis. Recently, we've just submitted this paper where we've team tagged with Rob Wood's group, where we're now extending this idea to now filamentous structures that program a little bit of the gait similar to worms, and we can start to see similar kind of locomotive gaits. Essentially, this is work, ongoing work, but we can kind of apply the same idea to soft filamentous robots. So let me go back here and see if there are any questions. I'm going to transition to a second story here.
That's the question of how does the wigglers coordinate? How do they coordinate with pullers?
Yeah, it's a very tricky question, and I don't have an easy answer yet. It's an open problem.
If you can you think
Yeah,
about it?
and I'll show you some preliminary work where we're approaching this problem through simulations. Yeah.
Can you suddenly raise the temperature on the cold side and suddenly cool down the hot side? And how quickly will they revert to their phenotypes if they will?
Good question. I haven't tried it. Yeah.
But in your model with the Dan's group, that's just a byproduct mutualism. There's no coordination. It's just the ones in the back are
Yeah.
flapping.
Yeah, basically. I'm not showing you other work, but essentially now that we had a platform, we could start to let it happen spontaneously, by which means we put a sensor and they would decide their gait based on the amount of light they would get. And we tried to evolve them, thinking about evolvable robotics. We did a lot with flexibility and emergent gaits. So there's a lot in the paper. I'm just kind of showing you the one key result, but there's a lot more we can play with.
But there's no internal.
No, they don't need to be. They can be asynchronous, if that's what you're asking. Beautiful question. So in living cultures, we can't, it's a very challenging experiment to one by one measure the worms. We do challenge some of our undergrads is to take worms and put them next to a rudal, because if you want to do kind of polydisperse versus monodisperse experiments, students complain and they leave the lab. So we try to leave basically a polydisperse culture and do these experiments, and naturally you get a distribution of these things. But think of it almost like a polymer with a stretch coil transition as a function of temperature. I'll show you some hint of early polymer, active polymer simulations where we can recapitulate the same idea very simple. Yeah. Yeah, and I'll show you a video. Yeah. Okay, so the next story here, essentially, the context here is that there is a young man, and he was, I think, 16 or 18, and he goes into this town of Gordium, and essentially the ancient folklore goes that whoever was able to, resolve this knot of rope tied on an ox cart would go on to rule all of Asia. And so when Alexander the Great came upon this, as all of you know, as the story goes, he took out a sword and kind of cut the Gordian knot. And really thinking about knots and tangles is both a historical problem, a mathematical problem, but something all of us deal with. We all at our homes and offices have a drawer which has a lot of USB or Ethernet cables that get tangled up. And so the question is, what can we learn from untangling from these living systems? And so if you watch these worms, which are in a shallow dish of water, this is real time. They really tangle up and form these beautiful tangles. And this is one of my favorite videos. I hope you can appreciate as they wind around each other, they start to form these braids and then they pull them in each other and they form effectively the formation of a blob here. And just to remind you, the blob is very cohesive. These are very interesting tangles. As you saw with Yasmin's video, you can hold them in your hand. They won't dissipate away. There is a lot of essentially resilience here in this structure. But Harry, a PhD student in the lab, discovered something while playing around with these worms in the lab. So here's in an aquarium, there's a worm blob in the corner. And he was trying to understand the response, and he's shown UV light on them. And he realized that within a blink of an eye, the whole blob, which is this cohesive tangle, completely untangled and dispersed, which was very surprising to us. It looks like the worms are exploding in 3D space. Here's a kind of closer look at this under a camera. And really just appreciate this. I'll play this real time and I'll slow this down. So don't blink. And you can see this cohesive blob completely unravel and untangle very quickly. And the two things that you saw coming up is a 9-volt battery wires attached to it. We're just giving them a little bit excitement. No worms are harmed in this, and they go back. We just have to excite them a bit. Here's it slowed down, and the answer to how they can untangle lies in this video. So I'll challenge you to watch this for yourself, track any favorite worms, observe their gait, and see if you can figure out the answer to the puzzle of how they untangle so quickly. Anybody care to venture a guess and spoil the puzzle? Say that again. Close, close. Say that again. Straighten, close. Okay, let me tell you the answer. So first thing, before we started to answer the puzzle of how they entangle, we had to actually quantify how this structure, how tangled was the structure. And here what we, after, I both love and hate this slide because it's about a year of work all encompassed in a slide. But effectively, after a long effort, we figured out that ultrasound, the same you use on when people are pregnant, effectively allows us. So here's an ultrasound probe from Philips, commercial one, and the worms are in this gelatin. We can actually scan these worms, and we can extract this data, which looks like this cloud of points. And with some segmentation and manifolding, really in collaboration with a team of people, we can actually, in XYZ, basically get the position of all of the filaments and resolve the tangle. So for the first time, we're peering inside this live tangle. And now that we have this information in collaboration with Vishal, who's a PhD student with Yon at MIT, but now he's the faculty at San Diego, we started to ask, well, how is this tangled structure? How do we describe this tangled structure? So here we're going to take two pairs of these worms or filaments. So if you take a blue worm and a green worm, you can see that they're touching each other, but they're not really tangled, same as these two worms. But you can see there are pairs of these worms, such as this blue and yellow worm and this green and orange, where they're starting to interlink with each other. And so to quantify this, we use a linking number. And an intuitive way to think of the linking number first, just for closed curves, is that if I have two curves and they're linked together, it's hard to separate them. This is a linking number of 1. And if I had two curves, but they're not linked to each other, I can separate them. So this is linking number of 0. We can apply this to linking number of open curves. And effectively, instead of getting this binary value of 0, 1, we can get essentially, with an approximation, something between 0 and 1. And we decide that if linking number is greater than half, these two filaments are linked together. And if they're not, they're just geometrically touching each other, but are not linked. And we can augment this with contact, because for two worms in a tangle to actually influence each other, they have to not just be linked, but also be in touch with each other, because if they're far apart, they might be linked. But what is important for us to also recognize is they're in touch. And so we augment this with a contact linking number. Same idea. If contact linking number is greater than half, these worms are touching and also linked with each other. The upshot of all of this is we can create a contact link matrix or a graph here where each worm in color, so this is about 12 worms, and you can see that each of the nodes is a worm, and each vertex that connects this is drawn when the contact linking number is greater than half. What this allows us to gather is that there are some worms that might only be contact linking with one other worm. There may be a worm that may be not linked with any other worm. But on average, with this graph, the mean degree of connectedness is about 2. And this is an important number that will play a role. What this says is, on average, each worm is interlinked with 2 other worms. And I explain, hopefully, in the end, the significance of this. Let me pause here and see if I can clarify anything so far. Said a lot of different things.
I have a simple question. When you untangle the worm with the proton, do they ever retangle the same way, the same manner that's right there, or do they retangle something different?
Very hard to answer that question. When I show the ultrasound data sets, we did 3 tangle data sets, and that alone took us about 8 months or so. A lot of this is it's an open problem to basically kind of draw these things. So a lot of this is semi-manual labor, and yeah, so it's very hard to do the same thing over and over again. But it's a great question. Yeah, this is multiple ultrasound data sets, experimental data of worms, frozen in space. So these are static at this point. Okay, all right. So the next step is now to start to move to dynamics, where we now want to ask about the untangling. And the first step we did is to develop basically an active filament model. Effectively, we're treating this as an active Kirchhoff rod. In this case, I'm showing you a video with the random head forcing. So essentially, the filament is moving. And already you can start to see it resembles something like a worm, but right now it's just a random forcing. To actually understand how these worms tangle up and untangle, we need to make a smarter, strategic, educated guess about the trajectories the head is taking and not just randomly moving around. And so to do that, we stared at a lot of worms when they both tangled up, as well as untangled up, and tracked essentially the head trajectory to understand, well, what are these gates? And this is what I was trying to ask you to do when I showed you the video. We want to understand, is there some special gate that they're adopting? And indeed, we found that the head gate, both for tangling and untangling, had similar motifs. And we can see this more clearly if I just play you one video of a worm, which I think some of you figured out. It does this helical motion. It goes kind of clockwise and then counterclockwise, clockwise and counterclockwise, and it switches the direction. And this is basically the same for both tangling and untangling. It's just the rate at which it switches changes. Hopefully that's clear. And so we made a simple couple parameter model where now I'm tracking the head as it turns, and the velocity is a tangential velocity over there. And if I track the head in blue as it does a right turn, then after a certain time, it's going to switch and essentially turn in the counterclockwise or left direction. And so there are 2 intrinsic time scales over here. One is kind of the 1 by alpha, which is essentially the turning speed while it is in kind of a turn. And then the second time scale is how fast or slow it switches. And so these 2 time scales kind of capture this problem, and we define essentially a dimensionless number called the chirality number, where we divide these 2. And here's an intuitive way to think about this. If this chirality number gamma is larger than 1, effectively the worm's head is tracing chiral loops. It's not switching its direction. But if the chirality number is less than 1, it switches direction and forms this figure 8. Everybody okay and so far? So this is now one number that helps us, I'm going to show you, basically controls tangling and untangling. Okay. And so now we have this for a simple filament. Now we need to create a stochastic differential equation and effectively be able to do these in 3D. So effectively, we write a differential equation for the turning rate of the head as well as the speed, where we have both 1D noise in here, and over here, S is essentially what's switching the head direction, the turning rate at some rate alpha, at some rate gamma from plus to minus 1. And effectively, we have the speed at which tangentially this filament is moving. And then all of this, if it was just happening in 2D, you wouldn't get any tangles. To truly form a tangle, you need to be in 3D. And so we add a little bit of 3D drift noise. So effectively, we just have a forcing function in third. So these filaments can raise their heads and they can tangle up. But effectively, they're pushed down so they don't veer off in 3D. So we're basically confining them a little bit, but they can still tangle and form a structure. And so there's noise in the forcing function as well. So here's what this looks like. On the left, we basically do large gamma. So this is effectively them switching less. And so you can start to see, started from a random configuration, they start to, voila, form a tangle as we hoped and expected. And on the right-hand side, if we start from an initial configuration that is reminiscent of the ultrasound, from starting point of the configuration from an ultrasound scan, and with a low gamma, we can see that this thing will untangle very, very quickly. And the bottom shows you the contact link tangle graphs showing both tangling and untangling. Let me pause here for a second and see if I have any questions before I go into the mean field theory. Yeah.
If there is an effect, certainly effect, but what is the magnitude of the effect of length of these?
Beautiful. So to effectively tangle, you need to have a critical length scale. If you're too short, like bacteria, you may not tangle. And too long, you may even form self-knots. And so that's something we're trying to do. Right now for this paper, effectively, we stayed about for the simulations at a monodispersed length and try to get kind of a be happy kind of match with the experimental thing. But there are definitely interesting questions to ask that. I'll show you maybe preliminary data of how we're approaching it. With swarm? Okay.
Big clump. It's very dynamic when you start tracking it. They're moving around a lot. I'm wondering when these are in the tangled state, are the worms not moving, or are they really moving around a lot in some sort of way that they're responsive to each other or something?
Right. So it's a little bit hard to track that because now we need 4D measurements of the worms if I wanted to see in space and time. But just observing under a microscope, you can see, and I'll show you one where we fluorescently tag some of these worms. You can start to see that they effectively, because they're confined, they're doing rotation and they can slide along inside and out. And they are doing that, but it's just a very challenging problem to track them. And they have to maintain the cohesive structure as well, because if they do that too fast, they, as you see, they dissipate away.
When they go from tangles to tangles, that's a very bad nervous.
Right.
How they're responding.
Yeah.
Have somebody actually figuring out what's going on with the nervous system.
Beautiful. A biologist, Charles Drews, actually proposed this lumbriculus system as a as a system to study neuroscience because it has a giant axon along its body, and effectively that's how it can transmit information and quickly react. And it has a lot of these longitudinal as well as circular muscles. So I don't have a good answer of how they do it, but they are a really unique system that other people in neuroscience also study them for understanding how the information along this giant axon or neuron travels. I'm not doing this, but it's a small community that does it on these worms.
I have a couple of questions. First is that the simulations are clearly sort of worms moving head first.
Right.
So driven by the head, is that true for the
Yes.
worms as well? Because they have not evolved yet.
Yeah, I haven't shown you the data, but we've done this. They're force followers. Their head is the only motion. The tail basically follows the same curvature.
The second question is so you earlier showed these transitions that they sort of spontaneously go through, in which they're blob and then they become lines.
Yeah.
Are they doing sort of the same thing in that, and what's driving those transitions?
Those are long time scale experiments, some of the time lapses as we observe them. But if you consider kind of the explosive detangling one, there's not enough time. It's happening in milliseconds. Is that what you're I'll show you some videos in which they're doing very bizarre behaviors, and it gets more complicated as you scale in. And if you have, instead of 50,000, couple 100,000 worm blob, I'll show you some phase separation stuff. But you're putting your finger on an interesting point, and it's complicated. I don't have a theory or answer yet.
So similar to that, try dinner experiments where you go from high gamma to low gamma and back and forth.
Yeah. Yeah, the title of the paper is reversible self-assembly of these things. And indeed, we demonstrated you can go back and forth. And I have one video that I'll show you.
One thing, I mean, in order to generate other structure.
Yes.
Back and forth. So
Oh, that's a harder problem. We can tangle, but there's control of these things is a whole different beast. So if what I described didn't make as much sense, we also make comics. So here's our lab comic, where in a page or two, essentially, if you wanted to save 45 minutes today and get it in 2 minutes, I'm going to pass around this comic and you can read. With every paper, we make kind of a 1 to 2 page comic to summarize our paper, because I used to think I'd publish a paper and then next day I'd wake up and I'd think that everybody would read it, and I'd see 1 or 2 downloads. It's just me clicking on it twice. And so I tried to make comics such that I know my child will never open up PNAS or science and read a paper, but maybe you can make it accessible for the next generation by making some comics. So anyways, okay. So the last part over here we wanted to think about is how do we create a predictive ability to go from worm head dynamics to understand measure of tangleness. And a common approach in kind of physics and mathematics you do is called what's a mean field approach. So if you have a tangle of these worms, if I chop them up and take a slice out of it and now create a mean field where each of these pegs are basically different worms, I just want to understand effectively what a single worm does against all these worms to be able to create some predictive mean field theory here. And so here's essentially what we've done is essentially think of all of these as other worms, but now in a much more controlled setting where each of these pegs are distributed as a different length L. And now I have a worm head trajectory basically winding around in this 2D playground in this mean field, and it can wind around these pegs, tangle, or it can unwind and untangle. And similar dynamics, I can define a chirality number. A loop number starts to become important here because the geometric aspects, the length, the speed play a role. And an intuition over here is that if the loop number is small, this head won't tangle around these pegs, but a larger loop basically allows it to wind around more pegs. And then we can define, similar to the tangling index, similar to the contact links, we can define a tangling index effectively suggesting the average number of points in which the trajectory mapped out by this head winds around to, which is the tangling, and then close to the contact linking. And so effectively, we're asking how many pegs does it wind around? And this is the analogy and analogous to in the mean field approach to the what I just showed you in 3D is how many worms a given worm is braiding with or winding around with. And so we can now generate this tangling index as a function of gamma and R, are 2 dimensionless numbers. And so here's some exemplars. So for R, loop number is same in both, but I'm going to change gamma again. Same idea. If gamma is larger, it's going to create these chiral loops and loop around more, and gamma is small, then it's going to unwind. And I can do these in simulations. Each of these squares is a simulation and make a plot, R versus gamma, where I plot the color as the tangling index. And you can start to see where low tangling index, where it doesn't tangle, is the blue region, and red is higher, which is more tangled. So let's walk through this one by one. So tangling index greater than 2, which is the white line is the 2, basically means very tangled structure. And untangled is basically tangling index less than 2. And then you have a critical tangling where each filament or worm is braided around 2 pegs or critically tangled with each other. And if you place all the experimental data on top of each other, we see that the untangling lies over here and the worm tangle lie here. And the takeaway here is that the worms control their tangling to untangling purely through gamma. They keep the R same. And there are some physiological reasons for why the loop number is invariant. So they basically change their switching rate to do this quickly untangling and move towards the lower gamma. There is a nice puzzle here about this critical tangling. And I said to you in the ultrasounds, we found that the worms, we found that each worm on average tangled with 2 other worms. And that's kind of where we drew this white line, where this is basically a trajectory. And if you watch what this trajectory or data point over here lies, so if you follow this trajectory of a head, it basically winds around this blue peg. It takes a loop and then basically takes a larger circle and effectively cancels it out. So here it did a counterclockwise and then it a clockwise loop. And so effectively it canceled it out. And so this is a nice, fun mathematical puzzle that mathematicians do, asking the question of how do you hang a picture frame around two pegs such that if any of the peg is removed, the picture will fall. Does anybody know the answer? Say that again. A paw? Oh, yeah. But, well, maybe not. Is that what you meant? Good. Okay. So effectively, you wind it around and then go around counterclockwise and untangle it such that now if you remove any of the peg, the picture will fall. It's not actually. And so effectively, this mirrors this critical tangling gate. And so what I'm trying to tell you intuitively is the worms are sitting at this critical tangle state such that it's almost the way they can quickly untangle. It's like a quick topological release mechanism. I'm going to stretch this analogy, just loosely give you the analogy that if I had a cloth made out of amorphous tangles, essentially it's like pulling one fiber and the whole thing falls apart. And because this is a survival mechanism for these worms, because fish eat them and leeches eat them, this may be an important thing for survival for them to quickly release. And so that suggests why the worms had a critical tangling. So this is kind of close to the end. I'm going to show you couple of videos of preliminary stuff that we're doing just to share with you exciting things. If you need to learn more, here's kind of the paper. And Ted, I didn't show you, but we actually can go and do this reversible tangle, but it's there in the paper. These worms, we've looked at thinking about active matter, polymer physics, thinking about the biology of these things. Recently, we discovered these worms not just move in air, water, but at the air-water interface. So you hear some happy worms in the bottom, but then we discovered that the worms can hang at the air-water interface and form what we call a buoy, which is functional and can locomote. So they're attached to the air-water interface with a tail. It just blows our mind. It looks like something out of science fiction. And this is what a worm buoy of 50,000 worms looks like. And it's a beautiful problem of thinking about how the tails at the 2D interface kind of confine the topology of these things. This is what worm buoys look like. They're like tiny Roombas. This is a bird's eye view of a thing, and you can start to see they form the buoy, and it's not just a pretty structure, it's functional. Harry's going to put some food, and you can see they suck up and assemble and can locomote and have functional features, and we're trying to understand what's going on. We're trying to understand kind of physics of how predator-prey interactions happen. Here's leeches feeding on worm in a blob. And we started to think about, well, how do predators feed on worms that are in a tangle? And we got an idea thinking about how we might untangle systems. So with a postdoc, Ishant, in the lab, we're thinking about how do we unravel kind of this Ethernet cable problem? How do we remove a cable? And we discovered something very interesting that we're referring to as topological resonance, is that if you shake a filament in a tangle at a particular frequency, it comes out at other frequencies, it doesn't. And it's very surprising to us that there should be some resonance in these systems. We've discovered that we did field work in Iowa, and we realized that the worms coexist with duckweed. And we realized that they use these as structures to hang on and even hang underneath in the duckweed roots. And thinking about how organisms can leverage environments to, for their function and use it as tools. Knots and tangles across length scales, thinking of them as living notoids. And now we have these bacteria, and we're working with Jake Soha to think about why snakes not. Really, it's opened a lot of different things. Here's a video about thinking about phase transition in worms. So this is a function of temperature. And Pratisha, a talented postdoc in the lab, is trying to model these as active polymers, and we're exploring tangling-induced phase separation in these systems. Hopefully, I've convinced you that worm blobs are cool and they do a lot of amazing things. And hopefully, you agree with me that they're a phenomenal system to think about topological active matter. And I joke that we've opened a can of worms. With that, thank you so much for your attention.